Now that we've talked about water, buffers, pH, the Henderson-Hasselbalch equation, we're going to start talking a little bit more about the details of structural molecules. And when we look at macromolecules like proteins and enzymes, they're made up of smaller things like your amino acids and peptides. And so what we're going to do now is spend some time thinking about amino acids and their various different characteristics. So part of those characteristics, we'll start off by talking about amino acids and how they really are these three-dimensional structures. They aren't just plain flat XY molecules, but they have a X, Y, and Z dimension to them. And I'll explain how that is. And then we'll follow up with some discussions about sections 3.2 about the structures and properties of amino acids, how they can act as both acids and bases, and then we'll talk about how those amino acids combine when they form a peptide bond, and how those peptide bonds are what hold together a string of amino acids in a full protein or a full enzyme. And then finally, my favorite part is that we'll talk about small peptides that have physiological activity and are interesting from the standpoint of biochemistry and what other small molecules can do in a biological biochemical system. So let's start thinking about amino acids in their three dimensions. So here's an example of a basic amino acid, right? And amino acids are just a fancy way of just really saying that you have something that has an amino group, which would be right in here, right? And that this amino group is really an NH2 functional group that I've just shown here. Or you can think of it as a NH3 plus if it's charged. Another important aspect of amino acids is really thinking about the carboxyl group. And here's an example of a carboxyl group. Okay. Carboxyl groups are what we call COOH functional groups that when they dissociate and give off a proton become a carboxylate anion known as COO minus. And we'll talk more about that a little bit later. But those are the two major components of an amino acid, both the amino group, right, and then the carboxyl group that we've shown here and here on the molecule. We can also see that as a ball and stick model, and then also in its true form, which is the three-dimensional tetrahedral structure, meaning you have a central carbon and then four things that are surrounding it that form a tetrahedral structure when you're just looking at an amino acid by itself. And then down below here we have a space-filling molecule where you can see the various different groups. So the central carbon shown here in the middle, the carboxylate group shown here, and then you have your amino group here, and this would probably most likely be something that might be your R functional group. The R functional group is really unique because this is what confers specific identity of the different amino acids from one another. And I'll talk more about the various different amino acids and their characters a little bit later. The other thing I wanted to point out is that when you're looking at the amino acid, that you have to also consider and take a look at that central alpha carbon that's at the very middle of the structure. So you have a central alpha carbon and you have these four things that are hanging off of them. Again, the amino group, the carboxyl group, a proton, and then an R side chain. And the R side chain is really important. And we're going to discuss a little bit more about that in the next section of our discussion when we look at the different activities and properties of these amino acids that exist in vivo. The other interesting property about amino acids is that they come as two stereoisomers, and that means they can be distinctively designated as either L or D amino acids. And when you took organic chemistry, you probably heard a little bit more about R and S. So the nomenclature in biochem, when you're talking about amino acids, is simply talking about an L and a D. So the equivalent would be R and S in organic chemistry when you're learning about the various different stereoisomers. And stereoisomers are shown here and here. They are basically mirror images of one another. So this is showing one amino acid, right? And then you're looking at another amino acid and its mirror image. So they are considered to be stereoisomers. They cannot be overlaid upon one another, which is one of the properties of amino acids. So down here below, we have an example of an L-alanine versus a D-alanine. So stereoisomers of the L-alanine and D-alanine, or just alanine amino acid. And one important thing to note is that most amino acids that humans use and most eukaryotic organisms use are in the L conformation, okay? Whereas D is used by bacteria, some prokaryotes, and other organisms. Although in the last 4 to 5 years, we know that D-serine is one amino acid that is synthesized by humans. And there are all sorts of ways in which plants can handle D amino acids. So it's interesting, you know, the derivation of where all these amino acids came from and why mammalian systems and human systems mostly only use L and only mostly synthesize L amino acids, while very few D are used by humans, whereas D is predominantly used by plants and bacteria.